So going into the sexual reproduction, because it belongs to the class Oomycetes, no? So these are the first class of fungi who can undergo sexual reproduction. Okay. And so it will take place through the development of Antheridia and Oogonia. Okay. Antheridia will produce the male and the Oogonia will produce the female. Okay. What will they produce? Antheridia, they will produce antherozoids, which are male, and Oogonia will produce the ovum, okay, or the egg, which is the female. Okay. So let's read. Sexual reproduction takes place by development of Antheridia and Oogonia, which are easily developed in cultures and also after death of the host when the fungus lives saprophytically. So developed in cultures means in the experimental lab, okay, you grew this fungus, okay. So you took you took a brush, okay, and then you took the fungus, let's say from the soil, okay. Then this is a Petri plate which contains a media, okay, nutritive medium, okay. It could be anything. It could be potato, dextrose, agar. It could be MS media. It could be different types of media. Then you just swipe it here and then you leave it for some time. You swipe it with the brush and then you cover it. Leave it for some time, after maybe a week or month. So that is your culture, okay. So you are culturing fungus, okay. So that is what we mean by culture, okay. It means when you let something grow and then you observe it in the lab. Okay, so you've understood what it means by the word culture. Okay, the species of the Baryonum are homothallic, okay, and some species are heterothallic. Means they have the same type of the vegetative, the vegetative body, and heterothallic means they have two types of mycelia, okay. Homothallic is one type of mycelia, heterothallic is two types, okay. Means their shape and are different, okay. Homothallic means the mycelium is all the same looking. Okay, again, how the oogonium will form? Again, there will be a swelling in the hyphae. Okay, so it could swell from the tip of the hypha. Okay, this is what the hypha. These are the multinuclei, okay, and cytoplasm, etc. That can swell, okay, like that. Or intercalary. This is the hypha. This is a swelling. And then the hypha, again, it continues. Okay, so they could, again, just like that development of the vesicle. Okay, so it will, the oogonium will develop the terminal or the intercalary swelling of the hypha, and it will become spherical in shape and cut off by a transverse septa from the rest of the mycelium. So the swelling happens and you will have a transverse septa separated from the rest of the mycelium. The antheridium, they will develop as terminal club-shaped swelling of the hyphal tip and may originate either from branches of oogonial stalk or from neighboring hyphae. Okay, so what this means is that this is the hypha, this is the septa, this is the oogonium. Sometimes it will come like a club, like this. Okay, it may come, the antheridium, it can come as a club. Okay, or this is the ovum and this is another stalk, okay, another stalk, another mycelia nearby, and it will come as a club. Okay, it can come like this also. Okay, so it can develop on the ovum also, yes, or it can develop from a mycelia also. Okay, where were we? Okay, so we are done with that. Monoclinous and declinous. Okay, so oogonial stalk or neighboring hypha. The antheridium is very much smaller in size than the oogonium, and a number of antheridia come in contact with the oogonial wall. Okay, then The young, do I go down or do I go? I think I go there. So the young oogonium is multinucleate and globose. Eventually, its protoplast is differentiated into central oosphere surrounded by periplasm. Okay. Okay, so let's look at first at the asexual cycle. So this one is the mycelium. Then the sporangium develop. No, remember at the terminal tip or intercalary part. Then from the sporangium you're getting a vesicle. No, and then remember, then the wall will start forming. Okay, and each of those wall will give rise to these what to say, what is this, zoospores? I'm sorry, it's zoospores. Then the vesicle will burst, it will release. Okay, then in the water they start swimming. When they swim and they get tired, they will encase themselves, okay, to cover from the elements. Then a germ tube will come. Where is it? Where? 1 minute, girls. Then a germ tube will come out, and then again the cycle continue. Again mycelium, again, so on and so forth. So the diagram is very easy. Okay, so when you mention about the asexual cycle, you've written, no? First we start with the hypha. Then at the tip is our intercalary position, the sporangium would form. Then that sporangium will give out a vesicle. Then inside the vesicle there will be cleavage, no? Then we will get the zoospores. Then the zoospore will come out of the they will rupture the vesicle and come out. Then they will swim for some time and encapsulate themselves. Then they will germinate and produce a germ tube, and the cycle continues. So in these points, 1, 2, 3, 4, 5, 6, 7, in 7 points you can write in short. Then you give the diagram. Okay, I gave you from this higher textbook because it's short and it's quick. Okay, because when it's so long, no, you'll not be able to finish to write. Okay, so let's look at the structure. So here, this is what was that? Ooplasm or periplasm. What was that word? Monoclinus and declinus. Okay, this is a monoclinus stalk and this is the declinus. So they are sticking to the wall. Okay, the antheridium sticking to the wall of the ovum. And then at the first, okay, so here you can see the oogonium also has many nuclei. Okay, it's multinucleate, those black dots. Then even the antheridium is multinucleate. You're seeing many black dots, right? So in both this is a multinucleate ooplasm and this is a multinucleate antheridium. Why it keeps slipping? Okay, then here they will concentrate in this region, all the nucleus. We call them as a periplasm. Then what happens? How many eggs form in, no? So we know there should only be one functional egg, right? Only one should be left. And here also there should be only one functional left. So what happens is that all these, they will degenerate. You get it? All these unnecessary nuclei will degenerate. Only one is left. Only one from here and only one from there. Okay, the rest will degenerate. Then this single one which is still left, it will undergo meiosis. Okay, so how is meiosis? From one cell, meiosis 1 happens, it will divide into 2. Okay, that's your meiosis 1. Then again, after meiosis 1, meiosis 2 will undergo. So these cells, they will undergo and develop into 4. No? So meiosis, from one cell we are getting 4 cells which have n number of nuclei. Okay. So what happens here? It was 2n. No? Okay. And now we are getting n. Right? N and 4 cells. Okay. Okay, let's start here. 2n, it divided, no? Okay. So again, this is 2n. Okay, again 2n. Then reduction will happen, and here you'll get n. Here you're getting n. This one also gives you n, and it also gives you n. So you're getting four what to say, megaspore mother cell or microspore mother cell. These terms, let me use a simple term. 4 pre-eggs or 4 pre-antherozoids. You getting? So out of these 4 pre-eggs or pre-antherozoid, 3 will degenerate. Let me draw betterly. 1, 2, 3, 4. You got 4, no? And these are all, they all were n. So out of these 3 pre-eggs and 3 pre-antherozoids, they will degenerate and leave only 1 functional pre-egg and 1 functional pre-antherozoid, which will develop into your egg, your ovum, and your functional male gamete. Okay. Again. Okay, there you see a functional female nucleus and a functional male nucleus which has traveled. Okay, this functional male nucleus, it has traveled via this fertilization tube. Okay, so this male antheridium, it will produce a fertilization tube which will pierce through the oogonium wall, and then it will release the functional male nucleus. Do you notice, girls, there are only 3 here? 1, 2, 3, and this one is 4. Okay, you're getting it, why they do only 4, no? Because of the one microscope mother cell which was left. Okay, remember I told you, out of all those multinucleus, all will die off, leaving only 1. And from this one, it will undergo meiosis to produce 4n. Okay, so you're getting 4. So that's why they have drawn only 4. Okay, so when you draw, also draw only 4 dots here. Okay, draw only 4 dots here. And egg also, at first it was 4 cells, but the 3 degenerated. Okay, so so here they are coming close. Okay, and the nuclei will start to fuse. When they fuse, they'll become the zygote or the oospore. Okay, let's pause here and see what they have written, otherwise it will be too heavy. So the young oogonium is multinucleate and globose. Eventually, its protoplast is differentiated into the central oosphere surrounded by the periplasm. Okay, so you can see this is the periplasm, the one without the nucleus, and the central oospore are those with the nucleus. Again, let's go down again. So both the regions are multinucleate. Okay, the number of nuclei in the oogonium, it will reduce with age, and then the surviving nuclei will undergo meiosis to form 32 nuclei. Okay, so there are not just one. I explained it in a simple term that only one survived, but a few they survive, and we should get 32 nuclei. Then from those nuclei, all will degenerate. All nuclei will degenerate except one. Okay, and that will become your egg. Okay, all nuclei except one in the oospore degenerate, and the remaining nucleus will form an egg. Okay, then